All right. Hi, everyone. You made it through your exam and you are still here with me. It is truly a Christmas miracle. So tonight, as advertised, I'm going to run through the AP Bio free response questions from 2019. What I do know is that there were two different versions of the free response questions, and I only have one of them because only one of them is being made public. And when I called College Board, it doesn't sound like they're going to make the other version public. So this is version O. So if you had version E, I apologize. This is not going to be any of the free response that you had, and I don't have access to those to go through with you. This is just version O. Okay, so here we go. Let's take a look. So I thought these questions were really great. I don't know how you guys felt taking it, but I thought they were really, really good questions, and I think there are definitely some good points to be had here. So I was trying to do this thing. I have a new Surface book. And I was trying to do this thing where I can write on the screen, and I don't think it's going to happen for me. I thought I had this perfect solution, but it doesn't transfer over into a screencast. So I'll work on that for next year's casting. But for tonight, I'll just verbally go through these. Okay, so question 1, auxins are plant hormones that coordinate several aspects of root growth and development. Indole 3-acetic acid, IAA, is an auxin that is usually synthesized from the amino acid tryptophan. Gene TRPT encodes an enzyme that converts tryptophan to I3PA, which is then converted to IAA by an enzyme encoded by the gene YUC. All right, so first point. So remember, each bolded word is a point. Circle one arrow that represents transcription on the template pathway. So you could have circled the arrow between gene TRPT and mRNA, or the arrow between gene YUC and mRNA. But it needed to be an arrow between a gene and mRNA because that is the process of transcription. The second part of part A, the second point, says identify the molecule that would be absent if enzyme YUC is non-functional. If enzyme YUC is non-functional, IAA should be absent. Okay, so those are your 2 points there. 1 point for the arrow between the gene and mRNA, and 1 point for saying that IAA would be the molecule that would be absent if enzyme YUC was non-functional. I'm just going to keep moving unless someone asks me to stop. It shouldn't take us too long, maybe 20 minutes to get through all these. All right, part B, predict how the deletion of one base pair in the 4th codon of the coding region of gene TRPT would most likely affect the production of IAA and justify. So your predictions should have been something to do with a frameshift mutation, or you could have explained a frameshift mutation by saying that all the amino acids after this deletion would change. Either of those would be fine for your prediction. And how the deletion, and how it would most likely affect the production of IAA. For this, you needed to say that IAA production would be inhibited, because if we don't have the enzyme TRPT, then we can't make I3PA, which means that we can't make IAA. So we would not be capable of making the precursor to IAA, which is I3PA, if we have a mutation in gene TRPT. Okay, so there's your prediction point and your justification point there. C, explain one feedback mechanism by which a cell could prevent production of too much IAA without limiting I3PA. So for this one, you needed to limit something in that rightmost column. So somehow you needed to regulate either enzyme YUC or gene YUC, something in that column, because those things are going to affect IAA, but they come after I3PA, okay? So you could say that IAA inhibits the production of more enzyme YUC, and that would be a feedback mechanism, negative feedback, where too much IAA comes back and actually inhibits its own production. So you could have explained something along those lines for part C. All right, part D, rhizobacteria are a group of bacteria that live in nodules on plant roots. Rhizobacteria can produce IAA and convert atmospheric nitrogen into the forms that can be used by plants. Plants releasing carbon-containing molecules into the nodules. Based on this information, identify the most likely ecological relationship. So this is mutualism, because the rhizobacteria produce nitrogen for the plant, and the plant produces carbon-containing molecules for the bacteria. Okay, so that's an example of mutualism in which both organisms are benefiting. Describe one advantage to the bacteria of producing IAA. I absolutely love this part. So we know why plants make IAA, but if you go back up to the top and actually look at what IAA does, how many more times am I going to say that? Then You can see that IAA is an auxin, which helps to with root development and growth. And if you look back up into part D, these rhizobacteria live in the roots. So it's beneficial because these compounds are going to help roots develop and the bacteria live in the roots. So you needed to mention something with that, using that extraneous information from above to really make a clear connection there. If you said a symbiotic relationship, that would not count because symbiotic relationships are going to be mutualism, commensalism, parasitism. Those are all symbiotic relationships. All that means is two organisms interacting. You did need to mention, I believe, as far as I know, the aspect that this is mutualism and both organisms are benefiting. I should have made this disclaimer in the beginning. Obviously, the scoring guideline is not out yet. If it was, we could just talk through that. So this is just my own analysis, right? So it's not going to be necessarily perfect, but it's going to be pretty close because I've looked at enough free responses in my day. Okay. All right. So those are the 2 points for part D. Part E, a researcher removed a plant nodule and identified, love this question. I love this whole question one. Identified several cheater rhizobacteria that do not produce IAA or fix nitrogen. All right. The evolutionary advantage of being a bacterial cheater is going to be that you benefit from the carbon-containing molecules from the plant, but you're not expending energy. So I'm sure they needed something to do with energy expenditure or not using as much energy to get that point because they are talking about an evolutionary advantage. Okay, so that's that first part. And then secondly, it says plants can adjust the amount of carbon-containing molecules released into the nodules in response to the amount of nitrogen fixed, predict the change in the bacterial population that would cause the plant to reduce the amount of carbon-containing molecules provided to the nodules. So here you're going to want to mention something like cheater bacteria populations increase, because if cheater population bacteria increase, that means nitrogen fixation decreases, and with less nitrogen, the plant is going to release less carbon-containing molecules. So that's probably what they wanted you to say for that prediction point. And that is question 1. So those are the 10 points from question 1. All right. We'll cruise ahead then to question 2. I like this question as well. Okay, so students studying 2 different aquatic plant-eating unicellular protist species, A and B, designed an experiment. And they designed an experiment to investigate the relationship between the 2. Okay, so they give us a bunch of information there. I'm going to skip over that just because I'm sure this is still fresh in your brain, or you can reread it if you forgot. So A says the growth curve for species B in group 1 and species A in group 2 have been plotted on the template. Use the template to complete an appropriately labeled line graph to illustrate the growth of A in treatment group 1 and B in treatment group 2. So if I scroll down quickly, you can take a look at the graph that you were supposed to put on here. This is where I really wanted to use my fancy new Surface and draw these graphs out for you, but it's just not going to happen. So I will verbally explain to you a couple different things. So your species A group 1 line should have been steeper than the species A group 2 line, okay? The species B group 2 line should essentially be a flat line. You needed your axes labeled, so you needed to have time on the x-axis, and you needed to say in hours. So you needed to have hours in parentheses there, and then just number of organisms, something on the y-axis. Number of individuals would have worked because that's what the title is to this table. But you did need to have both axes labeled. You would have needed to have hours, the unit labeled there. And then your interval is really, really important, and it needs to match what we see up here on this table. So with that, and this is where I wish I could draw, where both the species A and species B line ends, you should have had 70 hours. If you trace that down to the x-axis, that should have been your 70 hours. If you count by 5 hours, you will get to 70 at that exact line. So you should have had 5-hour intervals along the x-axis. And along the y-axis, you should have counted by, I think it's 100s, 1, 2, 3, 6, 7, 8, 9, 10. Yeah, it's 100s, which puts species A group 2, that top point, right at 1400. which is where it is on the table. So the tricky part there was you needed to match your interval to the lines already given to you, and then you could construct your species A line and your species B line from that. And again, the species A line was steeper. It goes up to 1500 and the slope was steeper. And then the species B line for group 2 is pretty much a flat line. Okay. Part B, as shown in the table, the student established treatment group 2 with 5 individuals of each species, provide reasoning for the reduced initial population sizes. This serves as a control of the total species in the population. So that's what you needed to mention here, because since we have 2 different species, 5 each, we still have 10 total species, and that's a really important control. So that's what you would have needed to mention for B. In C, it says the student claims that species A and B compete for the same food source, provide 2 pieces of evidence. So one easy one is saying when species A and B are together, species B is unable to survive. We can see that in the rightmost column there. The other thing that you could have said is when species A and B are together, species A grows at a slower rate or does not approach as large of a population size or something along those lines, indicative of the fact that species A is actually growing much, much slower when the 2 are grown together as opposed to when they're grown separately. So those should have been your 2 pieces of evidence there. Next, it says predict 2 factors that would most likely limit the population growth of species A in treatment group 1. This could be things like space. So it can't be anything involving species B because they are grown separately here. So space, nutrients, water sources, waste product buildup, any kind of like carrying capacity limiting reagent would have worked there. Anything besides like some sort of competition or something like that, because it's just species A there. So not a mix in that. Lastly, E says many protists contain an organelle called a contractile vacuole that pumps water out of the cell. Okay, out of the cell. Remember that water is going to be pumped more quickly towards environments with higher solute concentrations. And this says predict how the activity of the contractile vacuole will change under lower solute concentrations. So our contractile vacuole should be less active because there's less solute outside. So the outside environment is less hypertonic, and so water is going to move there less. Okay. Hypotonic. Okay. All right. Got through our 2 big questions. The rest of these are going to go so quickly. Any questions as I'm going through this? Mariah, what's up? Why would the contractile vacuole work less? It's going to pump less water out of the cell if the solute concentration is give me a second. Let me reread this. I just went through this with my students, but you could be right. Protist pumps water out. Okay, because so this is a really good question, Mariah. As far as I can tell, because it's the growth medium that has a lower solute concentration, the growth medium is a solid. Ooh, gosh, I don't know actually. Now that you mention it, my interpretation was the environment was becoming less hypertonic, but they don't explain the solute concentration of the protists. So that's slightly confusing. But because, in my opinion, because the growth medium it has a lower solute concentration, then the contractile vacuole is going to be working less. But it really all depends on the concentration of the protist in comparison. So let me table that one and continue to think about it. You could be right. Part B, sure, absolutely. And Mariah, I'll get back to you. I have to think about that for a second because it's the problem is like those environmental types are really all dependent on both groups, and we only are given one. So I gotta think about that again. Again, this is just me going through this with my own interpretation. It's not College Board's official report. Okay, so part B, as shown in the table. Oh, okay. So in treatment group 2, we have both species A and B together, and we have 5 individuals of each species. So that's a control factor because we have 10 total species in each tube or in each growing environment. What do they call it? Let's see. They say in a container. So it says in treatment group 1, the student placed 10 individuals of species A into a container with liquid growth medium and 10 individuals of species B into a separate container with an equal amount of the same liquid growth medium. Okay. In treatment group 2, the student placed 5 individuals of each species. So 5 individuals of A and 5 individuals of B equals 10 total individuals in that container. So that's a controlled variable. We have only 10 individuals in that container. And Mariah, once I'm rereading this, you are absolutely right. It's going to be less active that the contractile vacuole is going to be pumping water out because less water is going to be entering the protist. You're 100% correct now that I reread this question. And Kaylee, hopefully that helped as well. Okay, good catch there, y'all. Sometimes it takes a couple times going through these to understand what they mean. Trust me, I feel the pain that you guys feel during your exam. Okay, all good? I can answer more questions. Sorry, I didn't mean to just move ahead if people still have questions. Okay, number 3, the pyruvate dehydrogenase complex, PDC, catalyzes the conversion, oh, what a great cellular respiration question, of pyruvate to acetyl-CoA, a substrate for the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. Okay, A, identify the cellular location where PDC is most active. You needed to say the mitochondria for this. Pyruvate is made in the cytosol in glycolysis, but it then moves into the mitochondria to be oxidized into acetyl-CoA for the start of the Krebs cycle. So mitochondria was your cellular location. Part B, make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle. So for glycolysis, that should have no effect on NADH because this process of oxidizing pyruvate happens after glycolysis has already occurred because pyruvate is the product of glycolysis. The amount of NADH produced by the Krebs cycle, though, will be reduced greatly because we are not oxidizing acetyl-CoA, and that is an important reactant in the Krebs cycle. So we will be incapable of producing as much NADH. So stays the same for glycolysis, decreases. Yeah, that's like a trick question. I think they put it there to trick you because you're like, wait a minute, it's not going to affect glycolysis, but that can't be the right answer. But it is the right answer. It's not going to affect that at all. And the reasoning is what I kind of just explained because glycolysis happens before. NADH does need to be recycled. Yes. But it still shouldn't affect it as far as like they're still going to be able to recycle these in the electron transport chain. So it's not going to affect the amount that can be produced by glycolysis because it can still be recycled either through fermentation or through the electron transport chain, which it sounds like, that can still occur because the only reactant that we need there is oxygen and electron carriers. So if we're still capable of making electron carriers in glycolysis, then we would still be able to go through the electron transport chain, even if we're missing some of this electron carriers made by the Krebs cycle. Good question. C, PDC deficiency is caused by a mutation in a gene located on the X chromosome. A male with PDC deficiency and a homozygous female with no history of PDC deficiency have a male offspring. Okay, so you have a male with PDC deficiency, so they only have one X. Okay, so I really wish, okay, wait, here we go. This might be helpful. X to the H. I wonder if this will actually manifest. No, I was trying to do too much. So you have an X with a recessive allele on it and a Y, and you're crossing that with a female who's homozygous dominant. Okay, so when we make this cross, you should have seen that all of the female offspring were going to be heterozygous carriers, and all of the male offspring would just be dominant because they would get the one good X chromosome from mom, and they would get the Y from dad. So the probability that male offspring will have PDC deficiency should have been 0. The probability of any offspring having PDC deficiency should have been 0 because all of the females will just be carriers for it. So you should have had 0 for that. Any questions about 3 before we move on? Okay. 4. All right, acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron. A researcher is investigating the effect of a neurotoxin that causes the amount of acetylcholine to increase. Okay, Describe the immediate, note it says immediate, that's very important, effect of the neurotoxin on the number of action potentials in a postsynaptic neuron. That should be an increase, okay? That's not going to be an increase for forever, but in the immediate, it is going to be an increase because we have more neurotransmitters that are going to signal ligand-gated channels, which you can see in this diagram, that are going to lead to more action potentials in the immediate future, right? Eventually those will fill up and it's not going to lead to more action potentials. But in the immediate future, we should have an increase. Then it says predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. Maximum membrane potential does not change, okay? So that's going to stay the same. That doesn't change no matter what. They show it as 30 here. Sometimes it's shown as 40, but doesn't matter how many action potentials you have. The maximum membrane potential is always going to stay the same. And that's just like a nervous system fact. B, the researcher proposes two models, A and B, for using acetylcholine esterase, an enzyme that degrades acetylcholine to prevent the effects of this neurotoxin. In model A, acetylcholine is added to the synapse. In model B, acetylcholine is added to the cytoplasm of the postsynaptic cell. Predict the effectiveness of each proposed model and provide reasoning. Okay, so model A will be effective if you add acetylcholine esterase into the synapse. That is where acetylcholine is located. So you will see a decrease in acetylcholine, and that should be an effective treatment. Whereas model B will not be effective because if you throw acetylcholine esterase into the postsynaptic neuron, you will notice up there that acetylcholine never makes it into the postsynaptic neuron. It's just staying on the exterior. It's acting as that ligand for the ligand-gated ion channels. And so if you put it in there, it'll never see any acetylcholine. So that will not be a successful model. Questions about 4? Okay. 5, another good question. Okay. So for 5, the first part says, based on fossil data, the researchers estimate that humans, sorry, I'm going to move this a little bit, and their most closely related species in the data set diverged approximately 7 million years ago. Using these data, calculate the rate of DNA percent divergence per millions of years between humans and their most closely related species in the data set. So this was a tough point to get. For question 4, if you predicted it right but didn't, you still get a point for sure. Absolutely, Mariah. Sorry, I'm just reading your question now. Okay, so this part A is really a tough point because first you need to do a couple things here. You need to figure out which is our most closely related species. So you can see that that's the chimpanzee due to the fact that we only have 8.8% divergence in our mitochondrial DNA sequences. Okay, so that is the first step. Then we need to calculate the rate of percent divergent per million years. So you need to do 8.8 divided by 7, not 7 million because it says per million years. Okay, so 8.8 divided by 7, and you should get, I think it's 1.26% divergence per million years. Okay, so that's your answer for part A, 1.26, I believe is the answer there, 8.8 divided by 7. For part B, using the data in the table, construct a cladogram on the template provided. You should have had humans at the top, followed by chimpanzees, followed by gorillas, followed by orangutans. So humans, chimpanzees, gorillas, orangutans. The only one of those two that could be flipped would be humans and chimpanzees. Okay. And then it says provide reasoning. No, you can flip humans and chimpanzees technically because they share a sister taxon. So you actually can flip those and still get the points. Those are the only ones that, as far as I'm concerned, you can, because you see that most recent common ancestor, you can always rotate around one of those points. And it should not change, although, no, it shouldn't. I think you may still get the point. It's better if you had humans on top for sure, but I think you still may get the point. So I would hold out for that. But we'll see when the score report gets released or the scoring guidelines. Yeah, but technically, according to the laws of cladograms, if you rotate around that point, you're good. Okay, provide reasoning for the placement of Gibbons as the outgroup. Gibbons are going to be the outgroup because they have the highest percent divergence with every other species. And they're in the 18s, which is far and above any other species for percent divergence. So that makes them the outgroup. And then C, on the cladogram, draw a circle around all the species that are descended from the species indicated by the node within the square. My students got real mad about this one. It needed to be human, chimpanzee, and gorilla. So the first 3 lines needed to be circled. Even if you didn't have human, chimpanzee, and gorilla written on those 3 lines, if you have a circle around those 3 lines, you will get the point for that. Okay. But unfortunately you did need all 3. I know a lot of my students didn't circle the 3rd line. But if you like tilt this sideways and take a look at it that way, all 3 of those branches are coming out of that common ancestor. So you needed to circle all 3 lines there. Any questions about 5? Okay. 6, yeast. Okay. A, identify the role of, I'm not going to reread through this because I'm sure this is fresh in your mind. A, identify the role of treatment 1 in the experiment. Treatment 1 is going to be a positive control where you have all amino acids present. So positive control is what you would want to say for part A. Part B, provide reasoning to explain how mutant 1 can grow on treatment 1 medium, but cannot grow on treatment 3 medium. Treatment 1 medium has all amino acids present, whereas treatment 3 does not have methionine present. So you needed to say something along the lines of mutant 1, sorry, I just lost my train of thought. Oh, mutant 1 requires an external source of methionine or is not capable of producing its own methionine, something along those lines. Then for part C, yeast mate. Okay, so this is saying we are fusing mutant 1 and mutant 2 haploid strains to produce a diploid strain, and you needed to predict the results, okay, down here. Okay, so this is where I'm not quite positive what they're going to accept. So you definitely needed a plus in this top row, and then you definitely needed a plus for treatment groups 3 and 4, okay? Because they should be able to grow if we have both strains mixed and one's capable of producing methionine and one's capable of producing leucine, then we should be able to produce and grow in both of those environments when we mix. What I'm still debating is what you should have put for treatment 2. My initial instinct is to say if this no amino acid present and neither can grow with no amino acids present, that this would be negative. But then I'm also thinking to myself, well, maybe they just need a source of methionine and leucine, and those were both missing from treatment group 2, and now they have them. So jury's out on that one. I think we're going to have to wait until we see the score report to see whether or not you should have put a plus or minus for treatment 2. Okay. Any questions about that? My initial instinct was also to say plus, minus, plus, plus. But then the more I've stared at this question and thought about it, the more I'm unsure if they didn't expect to have pluses all down the line. But yeah, my instinct is also telling me plus, minus, plus, plus. So hopefully that's your answer there for you. Regardless, that would be an all or nothing point. So hopefully that is the answer that they're looking for. All right. Next, question 7. Okay, so this one I actually think is the hardest question of the entire exam. And this is my best guess. And I've talked to a lot of biology teachers, and we're all taking our best guess here. So it says A, based on the data provided, identify the gene that's most likely to encode a protein that's an essential component to glycolysis. My gut is telling me gene G, reasoning being that it's located in every cell type. So I'm relatively confident in that one. Again, we'll see what the score report says, but as far as I can tell, and any other bio teachers that I've talked to can tell, should be gene G because it's present in every cell type. Okay. Then for part B, the researcher observed that tissues with a high level of gene H mRNA did not always have gene H protein. Provide reasoning to explain how tissues can have no gene H protein. So here you want to talk something about gene regulation. Maybe we're regulating transcription here. Maybe there's alternative splicing that's creating a different protein. You could probably be pretty creative here. But something along the lines of gene regulation, like, gene H is inhibited in certain circumstances, and therefore transcription doesn't occur, something along those lines is going to get you your reasoning point. That one was a little easier to get than figuring out about gene G. But yeah, kind of a weird question. I'm assuming we're going to see some really low scores on that question, also because it's towards the end of the question set. All right. Last question. I actually think questions 7 and 8 were the hardest. Okay, so question 8, the petal color of the Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of petal cells. Petal color is determined by the pH. Okay. A, identify the cellular component in the model that is responsible for the increase in pH. So an increase in pH means we're getting more basic. That should mean we're losing hydrogen ions. So that should be the KH transport protein. Okay. Your reasoning why that or how that component is changing the pH is because it's pumping hydrogens out of the vacuole, which is going to make it more basic and less acidic. So we have proton pumps, we have those protons being pumped in, but then the KH transport protein is pumping those hydrogens back out, which is making the vacuole more basic, okay? And then B, a researcher claims that the activation of the transport protein causes the vacuole to swell with water. Provide reasoning. So you want to say that we're adding ions. We're adding potassium ions, and you can see that we have a lot more ions inside of the vacuole than we do on the left-hand side, which means that the environment has become hypotonic and the vacuole itself is hypertonic in comparison to its environment. So therefore, water is going to be drawn inside, or you could have said that water is drawn to where we have higher concentrations of ions. I'm sure any of those would get you the points for why this is swelling. Any questions about that one? Okay, cool. Well, those are the questions. If they ever like spontaneously decide to release the other ones, I'd be more than happy to do another session. We'll know more information once they start releasing scorings. Obviously, you guys will find out sometime between July 5th and July 9th or 10th what your score was, which is exciting stuff. But until then, put your mind at rest, and you should feel very proud because you conquered AP Biology. Right, so that's all I have for you tonight. Quick and easy. I'm looking forward to maybe talking to you guys over the summer with stuff, and best of luck in whatever AP classes you're deciding to do next year or in college if you're on that route, because that's really exciting as well. All right, y'all.